Hi, now I am going to discuss about applications of animal cell culture. Before seeing the applications, we should know about what is cell culture. The cell culture is the process of removal of the cells from an organisms and introducing them into an artificial environment which should be favorable for the growth of the cells. The applications of animal cell cultures are in various fields. For example, they can be used as a model system and toxicity testing, cancer research, virology, cell based manufacturing, gene therapy, genetic counseling, genetic engineering, and drug screening and development. First, the model system. The cell culture can be used as a model system because it offers you worthy models for studying the basic cell biology and biochemistry. The interaction between the disease causing agents and the cells can also be identified using the cell cultures. The effects of drugs on the cells can also be identified. The process and the triggers for the aging can also be studied by using the cell culture models, and it also used for the nutritional status. Next is the toxicity testing. Before we are going to introduce a drugs, we should test the toxicity in the cells. So the cell culture can be used for the toxicity testing process. By this, we can study the effects of newly identified drugs or cosmetics and chemicals on the survival and growth in a wide variety of cell types. Exclusively importance are liver and kidney derived cell culture. The cell culture technique play an important role in cancer research. The basic difference between the normal cells and the cancer cells can be closely studied by using this cell culture technique. It is probable to convert a normal cultured cells to cancer causing cells by use of the chemicals or viruses and radiations. Thus, the mechanism which cause these changes can be easily studied by using the cell culture technique. The culturing of a cancer cells can also helps to test a suitable drugs which is used to kill the cancer cells and the method for selectively abolishing the types of different cancers. Next, we can see the role of cell culture in virology. The replication of viruses in cell culture is one of the initial and foremost uses of cell cultures for the use in vaccine production. These cultures are extensively used in the clinical findings and isolation of viruses. It is also used in basic research to know how they grow and infect the organism. Next is the cell based manufacturing. The cultured cells can be used to yield many vital products. For vaccine production, large scale production of viruses can be done using this cell culture. For example, vaccines for chickenpox, polio, rabies, measles, and hepatitis B can be isolated by using this cell culture technique. To produce proteins with a medicinal or a commercial value can be produced in large scale by using these genetically engineered cells. For example, hormones, monoclonal antibodies, insulin, etc. are produced by using this cell culture technique. The culture cells can be used as a replacement tissues or organs for various purposes. For example, for artificial skin, for the burn treatment or accidental damage, and for ulcers. Next is the gene therapy. The gene therapy is an experimental technique which involves the insertion of cloned or altered genes into the cells by using our DNA technology to replace the defective genes which causing the genetic abnormalities or to prevent potential disorders. The main purpose of gene therapy are to deactivate the improperly functioning mutated gene and to swap harmful mutant alleles with the functional one by selective reverse mutation process, and to interchange the non-functional gene with normal gene by homologous recombination technology. And next is the to insert a new gene into the body to help combat a disease. Next application is the genetic counseling, the removal and culturing of fetal cells from a pregnant woman is the amniocentesis. It is an important diagnostic tool for the early diagnosis of fetal disorder. These cells can be examined for the abnormalities in their chromosomes and gene using karyotyping and other molecular techniques. Next is the genetic engineering. The cultured cells can able to reprogrammed with a new genetic material, that is DNA and genes. It provides a key tool for the molecular biologists to study the cellular effects of the expressions of these new genes, that is new proteins which are produced. So this technique can also be used to produce new proteins in large quantities in the cultured cells. Next is the drug screening and development. For a drug screening, the cell-based assay have become progressively important for the pharmaceutical industry. It is not only for the cytotoxicity testing but also very important for huge throughput screening of compounds that may have the potential to be used as a drug. The cell culture tests were done in 96 well plates, but in increasing use, it is currently being made of 384 and 1536 well plates. And thank you very much.